Hi, everyone. Thank you for joining us today for our webinar, Detecting Small Regulatory RNAs with Single Molecule Sensitivity and Single Cell Resolution using the microRNAscope assay. My name is Luke. I'm the webinar manager here at Bio-Techne, and I'm excited to welcome you to today's ACD support webinar. I'd like to thank everyone for attending today, and I'm excited to welcome our featured speaker, Eliot Smith. Eliot joined Advanced Cell Diagnostics as a technical support scientist in 2021, transitioning from academia with postdoctoral experience from Oregon Health and Science University and Stanford University, where he studied the roles of transmembrane channel-like proteins in the auditory and vestibular systems of the inner ear. Prior to that, Eliot conducted postdoctoral research at the University of Pennsylvania using zebrafish as a model organism to study the effects of NDR2 kinase on retina development and degradation. His doctoral studies were completed from the J. H. Quillian College of Medicine, where his research emphasized protein biochemistry and molecular biology to study human recombinant proteases. And with that, we will turn things over to Eliot. Go ahead and take it away.
Thank you for the introduction, Luke, and thank you to everyone for joining us today. Bio-Techne's microRNAscope in situ hybridization assay provides users with a powerful tool to probe the spatial biology of target microRNAs in the morphological context of cells and tissues. Today's webinar will provide detailed information about the microRNAscope workflow and highlight the results and applications of this assay. I will begin with an overview of the underlying RNAscope and microRNAscope technologies, followed by an illustration of some microRNAscope assay applications. I will then review the workflow in depth, including protocols for sample preparation and pretreatment, guidance on assay setup and appropriate controls, and troubleshooting tips. Then I will provide examples of customer successes from published research using microRNAscope before I conclude by answering questions from the audience. Please submit your questions at any time during the presentation. Representatives from our technical support team will answer questions via chat, and I will also answer questions at the end of the webinar. RNAscope technology yields advanced in situ hybridization assays with greater sensitivity than traditional in situ methods. Our assays use oligonucleotide scaffolds that we call amplification trees to achieve single molecule detection and single cell resolution. Many of our assays can detect multiple RNA targets on a single sample. The technology central to our assays can detect a variety of targets with different lengths using chromogenic or fluorescent detection methods for spatial mapping of RNA in cells and intact tissues. In the first portion of our assays, tissue sections or cells are fixed onto slides and pretreated to unmask target RNA molecules and permeabilize cells. Then, stable oligonucleotide probes hybridize to target RNAs. RNAscope uses pools of small Z probes that bind to the target RNA in pairs. These double Z probes form anchor points for the amplification trees. Next, the amplification protocol builds the scaffold in a stepwise manner. The result is large amplification trees coated with either alkaline phosphatase or horseradish peroxidase at the site of each labeled RNA molecule. This high concentration of bound enzyme leads to robust and specific signal development where double Z probes have bound. Upon signal development, you can examine the results by microscopy. These chromogenic or fluorescent labels allow for visualization of individual target RNA molecules in the form of punctate dots. Finally, manual counting or automated image analyses can be used to quantify the results. Specificity is built into our probe design. We can design a specific probe to your target of interest based on the accession number or exact target sequence. Our bioinformatics scientists channel this information through the proprietary ACD alignment algorithm to select the optimum sequence to ensure target specificity and uniformity of oligohybridization in the assay. RNAscope technology has applications across multiple areas of research, including basic sciences, infectious disease research, cancer and clinical research, as well as biopharmaceutical discovery and development. RNAscope applications include validation of single-cell RNA-seq data and spatial mapping of complex tissues. RNAscope assays can also incorporate immunohistochemical or immunofluorescent methods for combined RNA-protein co-detection. This table summarizes our portfolio of products. While RNAscope detects targets larger than 300 nucleotides, BaseScope can detect RNAs between 50 and 300 nucleotides in length. The microRNAscope assay we will explore today is a singleplex chromogenic assay that utilizes alkaline phosphatase chemistry and fast red labeling. This assay is available for the LicaBond RX automated platform and in a manual format. The miRNAscope assay has the capability to detect small non-coding RNAs and short oligos between 17 and 50 nucleotides in length, including antisense oligonucleotides, microRNAs, and small interfering RNAs. Like all of our assays, miRNAscope provides detection at single-cell resolution. The high sensitivity and specificity of miRNAscope improve detection of short target RNAs to facilitate data analyses. Users can also combine miRNAscope with immunohistochemistry for RNA protein co-detection on a single sample. Now we will discuss some applications of the miRNAscope assay. MicroRNAs can play a role in tumor formation by repressing the expression of tumor suppressor genes or indirectly promoting the expression of oncogenes. In neuroscience, microRNAs regulate gene expression during neuronal development. They are also implicated in pathogenesis of many neurological disorders, neuroinflammation, and immune regulation. MicroRNAs also act as positive or negative feedback regulators of immune cell activation pathways. Here we have an example of the sensitivity and specificity of the miRNAscope assay in sectioned mouse embryos at E15.5. The microRNA miR1243P localizes specifically in the brain during this stage of development. In contrast, a negative control probe targeting a scrambled sequence illustrates the lack of background in this assay. miRNAscope shows robust performance across species. Here we see expression patterns of miR1243P in mouse, rat, and human brain. Mouse liver is included as a negative control, as miR1243P does not appear in mouse liver tissue. These results show specific high-resolution detection of the microRNA miR1405P in mouse cartilage of developing digits, somites, and the inner ear. In contrast, the scrambled negative control probe reveals no background labeling in these samples. Here we see the expression patterns of multiple microRNAs in cervical cancer samples as well as head and neck cancer samples. The panels on the far right use a scrambled negative control probe to demonstrate probe specificity and lack of background. miRNAscope also has applications for studying small interfering RNAs as RNAi therapeutics. siRNAs can be designed to target specific oncogenes and downregulate their expression to suppress tumor growth. siRNAs are potential tools for the clinical treatment of acute brain injuries and neurodegenerative disorders. siRNAs can also target differentially expressed genes to mitigate the progression of autoimmune disorders. These miRNAscope results show successful delivery of a small interfering RNA designated siRNA1, as well as its abundance and distribution in mouse liver. The negative control specimen on the left was administered saline without siRNA. The specimen in the center received the treatment with siRNA1, which is targeted by the probe in this assay. The specimen on the right received a different treatment with siRNA2 to demonstrate the specificity of the probe targeting siRNA1. miRNAscope also detects the antisense oligos used in small oligonucleotide therapeutics. In the field of oncology, miRNAscope can demonstrate the target specificity of ASOs that can inhibit expression of cancer-promoting genes. Therapeutic strategies using ASOs can treat previously inaccessible drug targets, including targets implicated in neuromuscular and neurodegenerative disorders. ASOs can also be used to downregulate expression of key target genes to regulate immune cell infiltration in autoimmune disorders. In this slide, miRNAscope has been used to detect the abundance of a delivered ASO and its distribution in cardiac and skeletal muscle of treated mice. Negative control specimens were treated with a vehicle lacking the ASO. Now I will discuss the miRNAscope assay workflow. The miRNAscope protocol includes the same general workflow as our other assays. miRNAscope is compatible with FFPE samples, fresh or fixed frozen tissues, and cultured cells. The probe hybridization step uses universal pretreatment conditions regardless of the sample type. The signal amplification trees built during the miRNAscope assay afford singleplex detection using alkaline phosphatase and the fast red chromogen. The red signal can appear as punctate dots, as merged signal from multiple spots, or as cell-filling signal for very abundant targets. miRNAscope results are suitable for both qualitative and quantitative methods of data analysis. This assay is available for the LicoBond RX and as a manual assay. When requesting a custom probe to target a mature microRNA, we recommend that you collect some target information from the MIR-based database. On the right-hand side, you can search for your target. Here we have searched for HSA MIR205 5P. Once you have identified the mature sequence, please note the accession number or click to get the sequence. You will need this information when you submit the new probe request form. This is an example of the new probe request form. We have selected human for both the target gene species and the sample species. This is also not a xenograft design. We have indicated that this request is for a microRNAscope probe. The detection channel is S1 for all microRNAscope probes. We have included the target name as HSA MIR205 5P. We have included the accession number from miRBase, and we also had the option to submit the nucleotide sequence in FASTA format. The resulting microRNAscope probe is then given a catalog number and a name, in this case SR HSA MIR205 5P. The automated format of microRNAscope is designed specifically for use with the LicoBond RX system. You will need to acquire the microRNAscope detection kit and microRNAscope control and target probes from ACD. We also recommend ACD control slides to validate the functionality of the assay. The automated microRNAscope assay requires several reagents from Leica as well. These include the bond open containers, bond wash concentrate, bond DWAX solution, bond ER1 and ER2 solutions, and bond universal cover tiles. You will also need the bond titration kit, bond mixing stations, bond polymer refine red detection kit, and bond aspirating probe clearing system. The manual microRNAscope assay, however, uses ACD's pretreatment reagents and wash buffer, in addition to the microRNAscope detection kit and microRNAscope probes. The manual format also requires an image hydrophobic barrier pin, and again, we recommend ACD control slides for assay validation. The microRNAscope introductory kit includes all of these components except the target probe. As with all of our manual assays, microRNAscope requires the HiBZ2 oven and accessories for the assay to work as intended. We only recommend Superfrost Plus slides for our assays. Other assays can result in tissue detachment issues and are not recommended. You will also need cover slips for the slides. Other materials needed for the manual microRNAscope assay include tissue tech containers and slide racks, either a beaker and hot plate setup or a steamer for target retrieval, and common lab equipment such as a centrifuge and water bath. Other reagents you will need include freshly prepared ethanol, formalin, phosphate buffered saline, and xylene depending on your sample type. You may also want to prepare 5x SSC from a 20x stock solution. 5x SSC can be prepared ahead of time and stored at room temperature for up to 2 months. Only EchoMount and VectaMount mounting media are validated for use with microRNAscope. Other mounting media can negatively impact the results. You can use either 10% NBF or 12% formaldehyde diluted from a 37% stock for post-fixation during microRNAscope sample preparation. The nuclear counterstain protocol uses 50% Gills hematoxylin 1, and you will need 0.02% ammonium hydroxide freshly diluted from a 28 to 30% stock as a bluing agent to lighten the nuclear stain. Each sample type has varying sample preparation, storage, and sectioning guidelines. For FFPE, ACD recommends samples be fixed in 10% NBF for 16 to 32 hours at room temperature. Samples can be stored at room temperature with desiccants for up to 3 months and should be kept at 4 degrees Celsius with desiccants for long-term storage. FFPE samples should be sectioned at 4 to 6 microns. Fixed frozen samples should be fixed for 24 hours at 4 degrees Celsius with either 4% paraformaldehyde or 10% NBF. These samples can be stored at -80 for up to 3 months in an airtight container, and they should be sectioned to 7 to 15 microns. Fresh frozen samples should be snap frozen immediately after harvest. They can also be stored at -80 degrees Celsius for up to 3 months in an airtight container. Fresh frozen sections should be 10 to 20 microns thick. Cells can also be cultured using either cover slips or chamber slides and fixed for 30 minutes at room temperature. Cultured cells can be stored in 100% ethanol at -20 degrees Celsius for up to 6 months. I would like to briefly go over the workflow to prepare your samples for pretreatment. Please read the appropriate technical notes for your sample type for complete instructions. FFPE sections require baking and deparaffinization with 2 xylene washes and 2 100% ethanol washes. There is also an optional stopping point between the baking and deparaffinization steps. FFPE sections are then post-fixed at room temperature, either overnight in 10% NBF or for 2 hours in 12% formaldehyde. Following post-fixation, the FFPE slides are air-dried. Fixed frozen sections also require a baking step, which is followed by a 15-minute post-fix at 4 degrees Celsius in either 10% NBF or 4% PFA. Here there is also an optional stopping point after the baking step. The fixed frozen sections are dehydrated through a series of 5-minute ethanol incubations at room temperature, once in 50% ethanol, once in 70% ethanol, and twice in 100% ethanol. The slides are then air-dried. Fixed frozen sections then undergo a second post-fix at room temperature, this time either overnight in 10% NBF or for 2 hours in 12% formaldehyde. After this, the slides are air-dried again. Fresh frozen sections are fixed in either 10% NBF or 4% PFA for at least 1 hour at 4 degrees Celsius immediately after the slides are removed from storage at minus 80 degrees Celsius. The fixation time may need to be optimized for your samples. The slides are then dehydrated through a series of ethanol incubations, as for fixed frozen samples. The dehydrated slides are air-dried, and once the ethanol is evaporated, hydrophobic barriers are drawn around the sections. For adherent cell cultures, fresh cultures should be seeded and grown overnight to 80 to 90% confluency on chamber slides. When the cultures are harvested, disassemble the chambers and submerge the slides in 1x PBS. Cover slips can also be used. For non-adherent cell cultures, harvest the cells by gentle centrifugation. Resuspend the cells in 1% PBS to wash, and then centrifuge the cells again and aspirate the supernatant. Adherent cell culture slides are fixed for 30 minutes at room temperature in 10% NBF. Non-adherent cell pellets are resuspended in either 10% NBF or 4% PFA and incubated for 1 hour at 37 degrees Celsius. Once the cultures are fixed, they are dehydrated through an ethanol series. Non-adherent cells are initially dehydrated in suspension and then applied to slides with a cytocentrifuge. Following the dehydration series, you have the option to store the slides overnight in 100% ethanol. Adherent slides, but not non-adherent slides, are then rehydrated stepwise from 100% ethanol into 1x PBS. Finally, hydrophobic barriers are drawn around the samples. Once the samples have been prepared, proceed to the pretreatment steps. First, all samples are treated with hydrogen peroxide. FFPE and fixed frozen sections then require incubation in target retrieval buffer to recover RNAs that were cross-linked by overnight fixation. Hydrophobic barriers should not be applied to FFPE or fixed frozen sections until after target retrieval. Here there is an optional stopping point. The samples are then permeabilized with protease 3 or protease 4 in the case of fresh frozen samples. Incubation conditions for target retrieval and permeabilization are tissue and sample dependent, and I will discuss these conditions in a moment. Following protease treatment, proceed directly to probe hybridization. The probes are hybridized for 2 hours at 40 degrees Celsius in the high BZ2 oven. After probe hybridization, you have the option to store the slides overnight in 5x SSC at room temperature. This is the last optional stopping point in this assay. 6 AMP steps comprise the signal amplification portion of the microRNAscope protocol. These 6 AMP steps assemble signal amplification trees at the sites of probe hybridization and require use of the high BZ2 oven. Fast red chromogen is developed once the amplification trees are in place. The final phase of the microRNAscope workflow consists of nuclear counterstaining with hematoxylin, bluing of the counterstain with dilute ammonia, and mounting. The total runtime for this workflow is approximately 9 hours. As I mentioned, the pretreatment incubation conditions are tissue and sample specific. Most tissues require the standard pretreatment conditions. However, you should also consult the table in Appendix A of the microRNAscope user manual to find conditions for your tissue type. This table lists the incubation conditions for target retrieval in FFPE sections and conditions for protease treatment. The recommended target retrieval time for fixed frozen samples is 5 minutes, and cell cultures require treatment with dilute protease 3. Signal amplification and detection uses a multi-step procedure to assemble the amplification trees bound to the target probe. First, the probe binds the target RNA. Then the trunks of the trees are attached to the probe. Next, branches are added to the trees. Finally, the leaves are attached. These label oligos are conjugated to alkaline phosphatase, shown here as red dots. Once the trees are in place, alkaline phosphatase reacts with fast red to deposit the red chromogen, shown here as red stars. MicroRNAscope assays are also compatible with protein co-detection assays. ACD's RNA protein co-detection ancillary kit incorporates the steps of immunodetection into the core microRNAscope assay in our integrated co-detection workflow, abbreviated ICW. ICW uses a co-detection target retrieval buffer specially formulated to enhance protein epitope retrieval and block the tissue. After target retrieval, samples are treated with the primary antibody. A post-primary fixation cross-links the primary antibody to the target protein to protect against antibody stripping. The assay then proceeds with protease treatment, hybridization, amplification, and RNA detection. Following RNA detection, a co-detection blocker is applied to block all enzyme activity prior to incubation with the secondary antibody. This assay requires an HRP conjugated secondary antibody. The protein can then be visualized with either brown DAB or ACD's green chromogen. We suggest the green chromogen for enhanced contrast because the brown signal can be difficult to distinguish from the red signal in some cases. Finally, the ICW protocol ends with counterstaining and mounting. Here we see an example of microRNA protein co-detection in human cervical cancer tissue. Note the punctate red signal labeling the microRNA as well as the blue-green immunohistochemistry labeling. Now I will discuss the assay controls and scoring guidelines for microRNAscope. MicroRNAscope assays use a positive control probe targeting the small nuclear RNA RNU6, which is a housekeeping gene. The negative control probe is a scrambled sequence. Here we see the labeling of RNU6 in human and mouse tissues, as well as HeLa and 3T3 cells. In all cases, the positive control probe yields abundant signal, while the negative control probe produces no signal. Because microRNA targets are so small, individual punctate spots may represent multiple copies of the target. We have established a semi-quantitative rubric for scoring microRNAscope results based on signal abundance and distribution. We recommend using this semi-quantitative scoring method. Use of the control probes is necessary to control the quality of the samples and assay. The scrambled negative control should produce results with a score of 0, indicating either no stain or less than 1 dot per cell. A negative control sample with a score of 1 would not pass quality control. Similarly, the positive control should always give a score of at least 1, which corresponds to 2 to 10 dots per cell and either no or very few dot clusters. A positive control sample with a score of 0 would not pass quality control. Once you have confirmed the quality of your results, then proceed to score your other samples. A score of 2 corresponds to 11 to 20 dots per cell, with less than 25% of dots present as clusters. A score of 3 represents greater than 20 spots per cell and greater than 25% of dots in clusters. When doing image analysis, we recommend using ImageJ, QPath, or Cell Profiler. Some researchers also use Halo software to analyze RNAscope images. We have an RNAscope data analysis guide available online, as well as multiple recorded webinars that discuss analysis on these different platforms. Now I will provide some additional troubleshooting recommendations for common issues encountered during the assay. One critical step in microRNAscope is to sufficiently permeabilize the cells with protease treatment. For optimal results, the tissues should be neither underdigested nor overdigested. Nuclear morphology is used to optimize digestion conditions. Here we see an example of underdigested human lung adenocarcinoma tissue. The hallmark of an underdigested sample is the presence of nuclei with well-defined borders and no changes in nuclear morphology. In the middle panel is an example of desired permeabilization. While the nuclei maintain their overall morphologies, the borders are not as sharp as in underdigested samples. Finally, in the panel on the right, we see samples that have been overdigested. Nuclear morphology is abnormal, or as in this case, nuclei may not be visible with nuclear counterstain. tissue morphology may also be affected. In this example, the extended pretreatment conditions were optimal for these samples. This sample comes from mouse kidney tissue. Again, the underdigested sample shows virtually no digestion of the nuclei or changes in tissue morphology. In the middle, we see a sample with acceptable permeabilization. While nuclei and tissue morphology are generally intact, the borders of nuclei are less well defined. In the sample on the right, overdigestion has resulted in loss of both nuclear and tissue morphologies. In this example, the standard pretreatment conditions were optimal for these samples. Another common issue users encounter is sample detachment. Based on the sample type and pretreatment conditions, additional steps can be included to improve sample adhesion. Here we see the protocol modifications to solve FFPE sample detachment. For FFPE samples, if you are observing detachment prior to the postfixation, then add a baking step before and/or after the postfixation. If you are observing tissue coming off of the slides after the hydrogen peroxide or target retrieval steps, add an additional baking step before the protease step. All baking steps are for 30 minutes at 60 degrees Celsius in a circulating oven. In addition, ensure that you maintain a mild boiling of the target retrieval reagent to avoid bubbles getting trapped between the tissues and the slides. For sample detachment issues with fixed frozen tissues, we recommend drying the slides at -20 degrees Celsius overnight after cryosectioning, and adding a baking step after both postfixations for 30 minutes at 60 degrees Celsius. We have found that the majority of sample detachment happens after the target retrieval step, and so we recommend adding an additional baking step for 1 hour at 60 degrees Celsius before the protease step. After trying steps 1 through 3, if you continue to observe tissue detachment, then we recommend that you extend the protease time to 45 minutes and omit target retrieval. For sample detachment issues with fresh frozen tissues, we recommend extending the post-fix duration to 90 minutes at room temperature and including a baking step at 37 degrees Celsius for 30 minutes. If you continue to experience issues with detachment or other issues as mentioned earlier, please contact technical support for additional guidance. Many of our users have had success with the miRNAscope assay. Now I will highlight the use of miRNAscope in 2 peer-reviewed research articles. In this 2021 study from the journal Cells, the authors characterized changes in the microRNA profiles of rat retinal ganglion cells in models of nerve damage and ocular hypertension. Among their next-generation sequencing results, they identified significant downregulation of miRNA 181D 5P under conditions of increased intraocular pressure. They then used miRNAscope to visualize the downregulation of this microRNA in the spatial context of the rat retina, as shown here. They found that elevated intraocular pressure led to downregulation of miRNA 181D5P, specifically in the retinal ganglion cell layer and the internuclear layer. They also probed for LET7B5P as a control because its expression levels remain unaffected by increased intraocular pressure. Ultimately, the authors concluded that miRNA 181D5P elicited significant neuridogenesis in wounded retinal ganglion cells. In this 2021 article from Translational Oncology, the authors used transgenic mice with mammary-specific expression of the miRNA 200B, 200A, 429 cluster. They found that elevated expression of this cluster significantly reduced mammary tumor formation. They used miRNAscope to validate the abundance of the microRNA cluster and its specific localization to mammary gland tissue. The microRNA cluster was only present in the mammary tissue of the transgenic mice. In combination with other methods, the authors showed that overexpression of the miRNA 200B, 200A, 429 cluster inhibited expression of SAA1, SAA2, and SPP1, which are all elevated in mammary tumors. To summarize, miRNAscope enables detection of small RNA targets in situ with single-cell resolution using existing equipment. The miRNAscope assay supports research applications such as cancer, infectious diseases, neuroscience, developmental biology, and therapeutic target validation. This assay is available in automated LicaBond RX and manual assay formats with fast red detection. miRNAscope is compatible with the RNA protein co-detection ancillary kit for combining immunohistochemistry and in situ hybridization on one sample. The miRNAscope assay provides a robust technique to answer unique biological questions and advanced research in the context of spatial biology and transcriptomics. This webinar and past webinars are available on our website at the address shown here. To contact the technical support scientists, you can reach us by phone at 1-877-576-3636, via online chat, or by email at support.acd@biotechne.com. For sales inquiries, pricing, and ordering, please reach out to your regional account manager by email at acd_sales@biotechne.com. Thank you for joining us today. I will now conclude by answering questions from our audience.
Okay, and with that, I would like to welcome back our speaker, Eliot Smith, and we will begin our Q&A. All right, there he is. Welcome back, Eliot. Thanks for that great talk.
Thank you. Thank you again, Luke.
Excellent.
Our first question is it possible to use RNAscope system to detect the subcellular localization of ASOs, microRNAs, and small RNAs in cultured cells? You can use miRNAscope to label ASOs, microRNAs, and small RNAs in cultured cells. You can definitely distinguish between nuclear and cytosolic localization. And if you have a marker for your organelle of interest, then you can combine with immunohistochemistry in order to label that organelle.
Excellent.
Our second question, does the probe bind to the mature form of the microRNA or any of the previous immature forms? Example, premature microRNA, etc. So yes, the probe can bind the mature forms of the microRNAs. Typically, for most microRNAs, the mature species is more abundant than the premature species. But yes, it can potentially detect the pre-microRNAs. Another question, can microRNAscope red be combined with immunofluorescent staining? Yes, it can. Although I didn't mention it in the webinar, the fast red chromogen does have fluorescent properties and can be imaged with Psi-5 microscope settings. And then you can combine that with immunofluorescence.
Awesome. Next question here. Can I run, or excuse me, do I need to optimize my sample conditions for both miRNAscope and RNAscope if running in parallel?
No, you can use the same conditions for both assays.
Excellent. Is it
And
possible
I also,
oh, go ahead.
Sorry, go ahead. I also see another one here. Is it possible to run microRNAscope and RNAscope on the same sample? No, you cannot run these on the same sample at this time. You would need to run the assay on serial sections, would be the best approach.
Excellent. All right. We do have a lot of questions in queue that we'll be getting back with a text answer here. I might dive into a couple of them. So Eliot, if you can answer this one, if the tissue is already fixed, how does fixing it further improve microRNA quality?
It has to do with the stability of the microRNAs because they're so small. The extra fixation helps to retain those microRNAs so that you get better signal and more representative signal of the total microRNA population.
Excellent. And we'll keep going through as these keep coming in. RNAscope is promoted as a single molecule technique. What do you believe to be the sensitivity of miRNAscope in terms of molecules per cell?
With microRNAscope, because they're so small, you can't always rely on every spot being a single molecule. With our other assays, every spot represents a single molecule, but with microRNAscope, you can't. You're not necessarily guaranteed, and so we recommend either using that semi-quantitative scoring rubric or using like densitometry in order to quantify. but it's
Awesome.
not suitable for digital quantification.
Thank you. A couple more here. How can I be sure that staining is a result of binding to a specific sequence and not a random non-specific binding?
Well, a couple of things. First of all, there is also a positive and negative control probes that you use. The negative control probe is a scrambled sequence, and so that will demonstrate the specificity. Also, during the probe design, the algorithms that they use compare against BLAST and other sequence databases to confirm that there's no cross detection with other sequences. And then they will let you know if they do detect any kind of cross detection in their in silico design.
Fantastic. How does RNAscope scoring correlate with FFPE quality, frozen tissue quality? Can you speak to if/how RNAscope scoring will translate to NGS sequencing success?
So usually there's a good correlation with next generation sequencing. Of course, the RNA integrity of your sample is going to come into play. You can correlate all of this against the positive control probe, which is PPIB. No, I'm sorry, that's not correct. It's a microRNA. It's expected to have a certain amount of abundance in tissues, and then you can kind of use that to determine relatively the integrity of your sample.
Excellent. Last question in here. Can we use microRNAscope? Excuse me, I've just lost it there. Can we use microRNAscope for detecting microRNAs in liquid forms of our body?
No, it's only compatible with tissues and cultured cells and can't be used with like extracellular fluids and that kind of thing. You may
Excellent.
be able to do it on like red blood cell smears, but I'm not sure if that's been tried or not.
Gotcha. Thank you. All right. Well,